what I've been told, for many of their recreational waters, they have a system where they take samples multiple times a day, and they do rapid bacterial tests, and they'll post flags, green, yellow, red, at individual bathing beach locations to indicate what the water quality is. At least for the bathing beaches, yeah. To give you a complete case study based around going into a paper in the literature in detail. And you should have on your Piazza file not just the presentation, but the paper itself that I'm going to talk about. So I'll talk about the paper, the objective of their study, the approach, walk through the steps that Arun Kumar went through: dose response, exposure, risk characterization, and then discussions and conclusion. So this is the paper. It was published in 2013 by a group in the actually one of the authors, Graham McBride, is from New Zealand. The other authors are from the U.S., looking at a very specific question of what is the total public health risk to swimmers from stormwater exposure pathogens to the U.S. Now, in many parts of the U.S., including many coastal areas, we have a situation where stormwater runoff is often a dominant source of pollution. I think hydrologically somewhat dissimilar to India, but in addition to the point source urban outfalls, these non-point sources can be of significance. So they set forth an approach, estimate recreational water risk in waters impacted by stormwater sources. And instead of looking at indicators, as in the previous case studies, they looked at estimating risks attributable to specific pathogens that are known to be in the stormwater themselves. And to do that, and you were just introduced to this nicely in the last talk, you have to track what happens to the microorganisms from the point of release of a discharge into the water to the location where exposure to swimmers and other recreators occurs, and that means you have dilution and decay that could affect the level of exposure that individuals recreating in the water may encounter. They looked at dose-response relationships for the multiple classes of pathogens based on what they termed reference pathogens, which has been a widely accepted approach: viruses, bacteria, protozoa, that have dose-response curves developed in the literature. Some of these are in the wiki, some of these are from other sources. And then the third issue, and I think when Dr. Rose talks about enumeration, she'll mention this in a bit more detail. Classically, the way in which we've measured pathogens has been via culturing them. Petri plates or dilution tubes for bacteria, tissue culture methods for viruses, microscopy, and more recently, viable microscopy for protozoa. But for a lot of reasons, including the speed of the analysis, more rapidly we're moving toward molecular methods, PCR and qPCR. And viable methods don't always have 100% effectiveness at culturing all organisms that may be infectious. The molecular methods may give higher numbers in some circumstances because they only are detecting possibly fragments of DNA or RNA. So harmonization of data from those two different approaches for the authors to do this comparative study was necessary. So these are example dose-response curves. The viruses of interest, adenovirus, which is typically one of the more infectious viruses we encounter in water: rotavirus, norovirus, protozoa, Cryptosporidium and Giardia as the reference protozoa, and the bacterial pathogens down at the bottom represented by Salmonella. You also notice enterovirus is down very low as well. Most frequently, when you do virus analysis in water samples, unless you do some special techniques, you only are picking up the enterovirus. And so it's the other ones that are really intrinsically more infectious based on examining the dose-response curves. They looked at literature values and did experimental measurements for residential stormwater, that is stormwater from total residential communities, from communities that had commercial and light industrial activity, agricultural stormwater. CSO is combined sewer overflow. So in many of the older cities in the US, stormwater is collected into the common sewer that carries wastewater and then discharged either without or with treatment into the receiving body of water. So it's a mixture of both partially treated sewage and stormwater. Mixed-use stormwater from a diversity of land use types that can't be separated, stormwater from forestry lands, and then dry season urban runoff in fairly dry climatic conditions where you get peak storm events. And this was particularly done in Southern California. And I would suspect, although I don't know of a comparison that exists, the stormwater qualities in California in that period might be similar, Arun, I would think, to the onset of the monsoon rains that you have here in terms of stormwater discharges that occur. That's an interesting research idea, perhaps, for one of your students. On the dose side, so we have the issue of comparing various techniques: qPCR, microscopy for the protozoa, reverse transcriptase, RT-PCR, for a number of cases. And they developed a set of rules as how you convert one set of metrics to another. So, for example, for adenovirus 40 to 41, based on the literature that they examined, they assumed that one TCID50 tissue culture infectious dose for 50% of the aliquots cultured was equal to 700 genome copies in qPCR. So if you have genome copy measurements, if you have TCID measurements for adenovirus, based on the rule, you can pool those two sets of metrics together. And then they took the data that was available to them once they harmonized the different dose metrics to construct a probability distribution for concentration in the discharges themselves. Now, those of you who attended Dr. Gurian's boot camp yesterday, you've learned that a lot of risk assessment is describing the various inputs in terms of probability distribution. And so what they did was they looked at all the data that was available to them for Salmonella, Cryptosporidium, Giardia, adenovirus, entero, noro, and rotavirus, and they used it to describe what they term a hockey stick distribution. With microorganisms in the environment, whether it's water or air or effluent, in general, you get a skewed distribution, an asymmetric distribution. Rather than characterizing that by one of the common skewed distributions, let's say log normal, they instead used a simple geometric set of definitions explained in that sketch, where you have a peak concentration, half of the observations are below that peak, and then a certain portion are symmetric, essentially, to that low concentration, with a long tail stretching out to a maximum. So they use that data to calibrate what points A, B, C, and D on that curve were for each organism. Once you know what's in the discharge, you now have to ask how much do individual recreators ingest when they swim or do other sorts of activities. It's different for adults versus children. Children tend to swallow more water when they swim than adults. It's also different for swimming in salt water versus fresh water. This study was focused on marine discharges, so it's all salt water. There's different ingestions depending on the type of exposure. Yes, you may swim, but suppose you just wade or splash in the water. Or suppose you just go in the water up to your waist without immersing your head. Different levels of exposure for that activity. They estimated from the literature volumes that would be ingested in those various types of activities. And then combining the dose response information, the exposure data, as well as the dilution and inactivation data from the source to the point of exposure, they were able to construct probability distributions such as this for each of the stormwater types for various organisms. So this is the case for children with urban stormwater, primary contact, meaning swimming, and no attenuation occurring. So they're swimming right near the discharge point of the water. The various lines represent different microorganisms. The bacteria are at the bottom. The brownish symbol is given as N. I don't recall what the N and R, well, yeah, no, N is norovirus, R is rotavirus. So the two highest probability curves on this norovirus and rotavirus of illness to the children exposed to urban stormwater via primary contact without dilution or decay. And so they went through each of the types of exposures, each of the types of stormwaters for all microorganisms. Now, they omitted a number of factors in doing their Monte Carlo. They characterized the distribution of microbial concentrations very well, but they did not consider the uncertainty in the dose-response parameters. And when I give my dose-response talk later in the week, we'll talk about that in more detail. They also didn't consider variability in ingested volume. So, for example, and I don't remember the exact number that they used, but a common value for adult ingestion during swimming in marine waters is 10 ml per swimming event, but that has some uncertainty and variability associated with it. They did not put a distribution around that particular volume number. So, in fact, this distribution is too narrow. It should be broader when you consider the other potential sources of uncertainty and variability. But their conclusions from the study are that, first of all, there's enough data in the literature to construct this type of QMRA for a broad range of scenarios, exposure types, microorganism types, discharge types, to determine risk, particularly during and shortly after rainfall events. And I think there's been a consensus emerging, at least in the US, that wet weather events, exposures that occur within about 72 hours, 3 days of a significant rainfall, are those that pose the most risk. So this is really, for a near rainfall event, estimate that exposure. Under their analysis, the findings confirm that norovirus infection and possibly illness is the predominant risk during exposure. Not the bacteria, not the protozoa. And so, if we rely on indicators, we need to ask how good are the indicators at representing the potential risk of norovirus. And in fact, though it's good to hear that, there's a very strong Indian fecal coliform standard, which is in fact, if you're using 100 fecal coliform, 100 ml, that's a factor of about 2 to 4 below most water quality fecal coliform standards in the U.S. But the problem is in salt water, the coliform die off much more rapidly than virus. So it's probably underestimating the risk if this finding would hold true to the situation that was mentioned in Mumbai. But more research is needed in terms of the applicability of indicator systems as well as the generalizability of the finding that norovirus is the most significant agent. And then finally, the issue of harmonization of laboratory methods for different analytical techniques is really one that there's a lot of work needed. And for those of you, when we went through the introductions, I heard a number of you are experimental microbiologists. There's really a lot of work that's needed in determining how to convert from one measurement to another. And I think it's not a simple conversion factor. For example, it may be a different value for the ratio between qPCR genome copies and TCID counts if you're very near the source of discharge versus if you're 24, 48, 72 hours downstream of the source of discharge, because the rate at which viability decays is different, and we know that, than the rate at which the genome copy signal decays. And so there's really probably multiple PhD theses just in that topic alone that could be done. I think that's my last slide, and so be glad to take any questions. Yep.
Also, which indicate this is a predominant organism, the virus.
There have not, as far as I know, been similar qMRI studies outside the U.S., but certainly I think in Europe, from the papers I've heard over the years, there's a similar sense that norovirus is probably a predominant pathogen in swimming exposure there. Many of the illnesses that are found in the epidemiology studies that have been done in swimmers are symptomologically similar to what you would expect from norovirus. Okay, thank you.